Now let's continue for the today's topic. As I told you that this is the reflection that what we are going to learn today. Like suppose if you want to uh, make a insulin is a kind of drugs, right, which control the diabetes, right? So if you want to extract, make a insulin product for the market label, so how you will create that insulin? So you have to first isolate the desired gene from the human cell. So that is your desired gene of insulin that you have to incorporate with the bacterial plasmids. That is called recombinant plasmid, which we have already learned. Now this recombinant plasmid, now you have to insert into the transgenic bacteria. Okay, so after inserting this transgenic bacteria, so this become a transgenic bacteria, and this transgenic bacteria you have to grow in the culture. Culture means not in the petri plate, but cultured in the a huge what we can say the bioreactor. Because a huge bioreactor facilitate the all the basic requirement, the condition, optimum temperature, everything is supplied in that container. So the bacteria can able to produce the ample amount of insulin that you have to isolate, extract it, and that can be considered as your insulin product, and then that product can be launched in the market, so it can be easily accessed to any what we can say the patient, right? So how this insulin, what are the process are there that we are going to focus today, started from the isolation till their product formulations. And we also learn that what are the bioreactors and what are the downstream process. We also focus on that. So let's move ahead and try to understand. Now this was just a recall what we studied about, that is related to the plant, like all the transgenic plants, right? We have already studied the transgenic TI plasmids, tDNA, different type of vectors. We learned about it, right? So what are the role of that bacteria? So you can see here is Agrobacterium tumefaciens, is a kind of soil bacteria which is present inside the soil. So so that Agrobacterium, the vector can be used TI plasmids, and that TI plasmids become a recombinant DNA TI plasmids that incorporate with the plant cell. So the plant with the new traits, or we can say it's resistant to the disease or different kinds of infections. So these are the steps we have already studied a little bit about the basic concept in the previous sessions, like previous 2 sessions. Now today we will focus that how to isolate a particular desired DNA or RNA from the any cell. Like you have a cell, then how you will isolate the DNA from that cell. So let's focus on. So this is the real concept, like how we can make vaccines. This is a AstraZeneca vaccines, and how it is produced, that we will see. So the first step of process of recombinant DNA technology is the isolation of genetic material. Now how you will isolate the genetic material from genetic material in the form of DNA from any particular cell. Like suppose if I give you a cell and I told you that you have to isolate the DNA from this cell, how you will isolate? Is there any what we can say the simple process that you can with the help of needle just extract the DNA? No, it takes a very sophisticated techniques to isolate the DNA. What you will do? So first of all, you should know that where is the DNA present. Yes, where is the DNA present? Now what is DNA? DNA is a nucleic acid. Yes or no? Yes, nucleic acid, like DNA and RNA, is the genetic material of all the organisms. Like if they are the multicellular organisms or unicellular organisms, all type of organisms, they have their nucleic acid, either DNA or RNA. In virus it is RNA, right? So DNA is the majority of DNA is act as a majority of nucleic acid in most of the organisms. So for cutting the DNA with a restriction enzyme, it need to be pure and free from other macromolecules, because DNA is, when you extract the DNA from any particular cell, that doesn't mean that you are just extracting the DNA. Apart from the DNA, there are certain others macromolecule will also come, like proteins or RNAs will also come, right? So these are the other macromolecules which is not required, which is not the part of our experiment. Our part is to isolate the desired DNA. So how you will isolate? So for that, DNA is covered by the membrane. So it is break the cell open to release the DNA and other macromolecules like RNA and protein, polysaccharides and lipids. Right? So to extract the DNA, it is obtained by treating the bacterial cell or the plant cell or the animal cell with the very specific enzymes. Like suppose if you have a bacterial cell, if you have bacterial cell, so what type of enzymes you are going to use? Yes, tell me. If you have a bacterial cell, then what you will use? The name of enzymes. Anyone? Yes, very good. It's a lysozyme. Okay, lysozyme is the enzymes which is used to break the bacterial cell. If there is a plant cell, if there is a plant cell, then what you will use? Cellulase. Very good, Bhuvana. Yes, cellulase. Cellulase. Right? Why? Because bacterial cell is made up of from what? Tell me. Why you need to use lysozymes? Yes. Responded fast. Glycogen. Oh yes. So it is made up from glycogen, or we can say polyglycogen, right? Polypeptido. Yes, it's a peptidoglycan. So that's why we need to use lysozymes. Plant cell is made up from cellulose, and that's why we need a cellulase to digest the cellulose. Similarly, if there is a fungal cell, okay, if there is fungal cell, fungus cell, then what you will use? Chitinase. Chitinase, because it was made up of, because it is made up of from chitin. Fungal cell wall is made up of from chitin. Very good, right? So that's why you need to first of all identify what type of cell is this, whether it is a bacterial cell or whether it is a plant cell or fungal cell. So based on that you have to identify, and then you have to use the desired enzymes. Now as we know that genes are present on long molecule of DNA which is intertwined with the proteins like histones, RNA, right? Because DNA is not a a single or separated. It is mixed with a lot of macromolecules. Like there is a DNA, RNA, some proteins, some histones are there. So lot of they all are combined together. So how you will separate all these things? So to remove the RNA, suppose you extracted here is the extracted DNA, extracted genetic material, but in that material there are a lot of proteins, some other macromolecules are there. So to remove the RNA, because your desired is not RNA, right? your desired is DNA. So RNA can be removed by treating with the ribonuclease. Yes, if you want to remove RNA, you can treat that. Treat means you have to add the ribonuclease. Ribonuclease. Ribonuclease is an enzyme who going to denature the RNA. Similarly, if you want to remove proteins from that macromolecules, you need to use protease. For protein, it is protease. Protease, right? So here you can see the cell content are treated with the protease. Why protease? To destroy the protein, and RNase to destroy the RNA. Clear? Now, your macromolecules, your genetic material is pure in the form of DNA. Right? Now this is all separated, but it is not separated very easily. So you need to do a centrifugation. So here you can use the centrifugation. Centrifugation is a kind of techniques. You can separate the two molecules, heavy and lighter molecules. So the cell debris, what is mean of debris? That all the RNAs and protein which is denatured, that is a cell debris. Okay, so that cell debris is pelleted in the centrifuge, and then the supernatant containing the DNA is transferred to the clean tube. So here you can see the red one is your debris, which is not your required product, but the yellow one is your DNA, which is your required product, right? So that you can easily, because these debris are heavy in weight, so it is settled down at the bottom, right? If it is settled down at the bottom, it is in the form of pellets, okay? And the DNA is present in the liquid. So you have to just easily transfer that liquid into the another tubes, and that tube exhibit a complete DNA of your desired. So the DNA is precipitated with the ethanol, and it form a viscous strand that can be spooled in the glass rod. So see, after transferring it, you need to add a chilled ethanol. Okay, so the purified DNA finally precipitated out after the addition of chilled ethanol. You have to keep in mind what chilled ethanol. Chilled ethanol is required. So you have to add the chilled ethanol, and then you can transfer the entire supernatant into the another tubes. That can be considered as your pure DNA. Is this clear how to isolate the genetic material? Yes, all of you please respond it. Okay, responded fast. Only 3 students responded. Yes, Anthony, are you there? Okay. Now the next one.
Yes, sir. Yes,
Good.
sir. I'm live, sir.
Okay, on your front camera also. So this is the first step, that is isolation of genetic material DNA. Now the next step is cutting of DNA at a specific locations. Now after isolating, your DNA is like a long thread-like structure. Okay, once you isolate it, the DNA is somewhat like long thread-like structures. Now how you will find out, how you will cut this long thread-like structure DNA at your desired location. So this is your DNA. Okay, So you need to cut it. So for that, under the optimum condition, the purified DNA is cut by the restriction endonuclease. Okay, you need to cut by the restrictions enzymes. And after cutting these restriction enzymes, agarose gel electrophoresis is used to check the progress of restrictions enzyme digestion. Okay.
Sir,
Yes. Yes.
sir, is that isolated DNA is a pure, like purified DNA?
Yes. See, this is your purified DNA. Purified means now this DNA is free from other macromolecule, because your interest is to isolate the desired DNA, genetic material. You need a genetic material. Okay,
Yes, sir.
but protein, then histones, then RNA, because if you're doing in virus free or like any bacterial cell, so virus RNA is not a genetic material for bacteria, right? So that is your macromolecule which is not required, so that you have to remove it, right? So how to remove? By treating the different enzymes so that it will easily get digested. So only DNA is remain in that, what we can say, the supernatant. So that is your DNA, a pure DNA. Is this clear, Sasidaran?
Yes, sir. Yes, sir.
Any doubt in this?
No, sir.
Okay. Now this is what? This is the DNA which you have isolated. Where you have isolated? In this tubes. This is your pure DNA, which is a long thread-like structure which I drawn over here. Okay. Now you need to cut it with the specific location. How you will decide this specific location? So you can use a restrictions enzymes. To cut this specific location, you need a restriction enzymes. Restrictions enzymes. Now these restrictions enzymes, when you add this restriction enzyme in a tube, like suppose this is a tube, and inside when you add the restriction endonuclease enzymes, you need to transfer this into the agarose gel electrophoresis. Why we need to transfer this into a agarose gel electrophoresis? Because this electrophoresis is going to help you to check the progress of restrictions enzyme digestion. What kind of, how this enzyme is working on digestion of that DNA. Since you know that DNA is a negatively charged molecule and it moves toward the positive electrons, right? So this process is also repeated with the vectors also. Like suppose you selected the DNA and you also select the vector. So vector is also passed through the same process. Vector of your desired vector that you need to transfer into the agarose gel electrophoresis to check the progress of their cutting, because when you isolate the vectors, vector is also need to be cut on into the locations, right? So for the cutting we need to add into the restriction enzymes, right? So after adding DNA and vectors together in the gel electrophoresis, it cut the DNA as well as vectors at this specific locations, right? So after cutting, the next is formation of recombinant DNA. So after cutting, formation of recombinant DNA. So in that cutting steps, the next is formation of recombinant DNA. So how this recombinant DNA form or rDNA form? So after the cutting the source of DNA, the vector DNA with the specific restriction enzymes, the cut out gene of interest from the source of DNA and the cut vector with the space are mixed and the DNA ligase is added. That means of this is your gene of interest, and this is your vector that you want to incorporate with this, right? So when you add this, you need enzymes. What is that name of enzymes? It is called DNA ligase. So when you add this with the help of DNA ligase, this is called the formation of rDNA, or it is also called the hybrid DNA. This is called the rDNA. You can call it as a hybrid DNA, or you can call it as a chimeric DNA. Yes, it is also called the chimeric DNA. So please remember this different name. What is rDNA, what is hybrid DNA, or what is chimeric DNA? Is this clear to all of you, the second step? I want a response from everyone. Quick response. Okay. The next is amplification of gene of interest using PCR. Amplification of gene of interest using PCR. Now here, what is mean of amplification of gene using PCR? So that means of the polymerase chain reaction, or the PCR. PCR is called the polymerase chain reaction. Polymerase chain reaction. Now what is this polymerase chain reactions? It is commonly called it's commonly called PCR, and the full form is polymerase chain reactions. Was originally invented by Carey Mullis. Please remember the name of the inventor. PCR, that is called the Carey Mullis. Carey Mullis in the year of 1985, who invented this PCR techniques. Okay. Now this Carey Mullis shared the Nobel Prize with the Michael Smith. Okay. Carey Mullis received Nobel Prize, sharing basis Nobel Prize with the Michael Smith in the year of 1993. And the best way to describe the PCR, what is PCR? Simply we can say it is the DNA replication in vitro. It is PCR is what? It is a DNA replication. DNA replication. You know the meaning of replication? Multiply, right? Means if you have only one gene of interest, you can make n copies, n number of copies of that DNA interest of gene. That is what called replications. So replication, DNA replication in vitro is PCR. In vitro is called PCR. Here, okay. Now, the basic requirement of PCR. What are the basic requirement to conduct the PCR? So to perform the PCR, you need three basic requirements. The first one is a DNA template. What is mean of DNA template? That is your desired gene which you want to use for making the product, right? So that is your gene of interest. That is your DNA template. Then two nucleotide primers, means DNA, you need a DNA template, you need a primers, and along with that you also need an enzymes. Okay, these are the three important what we can say, the basic requirement to perform the PCR. Okay, the first is DNA template, that is important. Then for primers, 2 primers you require. Why we needed 2 primers? Is it okay with the 1 primers? Tell me why we needed 2 primers. If it can work with the 1 primer, then it's all right. Then why we need to use 2 primers over here? Yes, primers are unidirectional, right? So DNA template is what we can say, it is bidirectional. So when you cut that, when you denature the DNA, this DNA is always double stranded. So when denatured, it becomes a single stranded. So single stranded means what? 2 bands are there, which is separated. So 1 primers you need to anneal over here, 1 primers over here. So it goes like this way and this goes like this way. That's why it requires 2 primers. Is this clear? Yes. So therefore it requires 2 primers and the specific enzymes. Now here you can see, when I say DNA template that contain 1 or more target DNA molecule to be amplified can be taken as a template. Like your gene of interest is right, so that you can take either one particular gene or particular DNA, or you can take multiple copies of the same, right? Another things you required is 2 primers, because the primers which are oligonucleotide that hybridize to target the DNA region. So 1 to each strand of double helix are required, and that's why, because DNA is a double helix, so double helix, 2 primers required. Now these primers are oriented with their end facing each other, that allow the synthesis of DNA towards one another, right? And the another one, the last one is the enzymes. You require DNA polymerase, because you are working with the DNA, which is a part of replication. So you require DNA polymerase. DNA polymerase. Okay. Now this DNA polymerase, which is stable at high temperature, it is stable on high temperature, like greater than 90 degrees Celsius. Okay, and is required to carry out the synthesis of new strand. So DNA polymerase, like what type of DNA polymerase you can take? Like tag polymerase. Okay, you can write the name, that is called the tag polymerase. Please remember this, tag polymerase. Why tag polymerase? Because tag polymerase is generally used in PCR reactions, which is isolated from a very popular bacteria which can survive at high degree temperature. Like we can say, the name of that bacteria is called Thermus aquaticus. Thermus aquaticus is a such kind of bacteria who can survive at high temperature, right? So I'm just writing the name, that is called the Thermus aquaticus. Yes, over here. Thermus aquaticus. What is this Thermus aquaticus? It is a bacteria. It is a bacteria through which the tag polymerase is generally used in PCR, which is isolated from the bacteria Thermus aquaticus. Other thermostable bacteria polymerase can also be used, but this Thermus aquaticus is most common, and that's why TX. See the name, Why it is called T? Because capital T is a thermos and AQ stand for aquaticus. So that is a tag polymerase. Is this clear? It's very easy for you to remember. Tag polymerase. What is tag polymerase? It is a kind of what? Tag polymerase is what? It is the polymer. It is the DNA like. It is a DNA polymerase like a tag polymerase, which is generally used in PCR process, right? Which is isolated from the bacteria, and that bacteria is called Thermus aquaticus. So now this is enzymes. Now let's learn the working mechanisms of PCR. What is the working mechanism? So how actually PCR work? PCR is what? It is a kind of instrument. You can see here. This is the PCR, a kind of very small compact instrument. It is generally not found in a very what we can say simple lab, but it is found in like various institute. Like one of the famous institute is Agarkar Research Institute, which is in Pune. Okay, Pune, you might have know of Pune. What is Pune? Pune is a place in Mumbai, in Maharashtra. Yes, all of you know about the Pune. Yes, so in Pune there is a one central government institute which is called ARI. ARI. Okay, so ARI having this instrument which is called PCR. So it is just a very compact instrument. You can take, you can transfer very easy from one place to another place. It's a very simple, like laptop we can say, or we can say a kind of a small Xerox machine. You know, Xerox machine at home we have just a small compact Xerox machine, similar kind of this PCR. Now, what is the working mechanism of this PCR? PCR basically work on three basic simple steps. Now, in that PCR, one single cycle follow three basic steps. The one basic step is called denaturation, annealing of primers, and then synthesis, which is called, so this is third one, this is called extension. Okay, these are the three important steps. First is denature. Please always remember, sometimes you know in NEET exams they ask only the, they will ask about the sequence. So sequence you should know. First is denature or first is annealing of primer. So first is denature, second is annealing, and the third is extension. Okay, this is the three important steps: denature, annealing, and extension. Clear? Now let's try to understand what is denatured. So when I say denatured, that means of melting of target DNA. So any target DNA, like suppose any your desired DNA, that you need to first of all melt. Melt means what? It is double stranded. So you need to separate this double stranded into single stranded, right? To separate the DNA is a denature. But it is not like that you can just hold with the two hand and trace like this. No. What you have to do? You have to just keep a high temperature so that this DNA gets separated from double stranded to become a single stranded. Right? So for that purpose, the denaturation steps, the target DNA is heated at high temperature. What is the temperature? That temperature should also be noted. The temperature is near about 96 degrees Celsius. Okay, so but the most common we can say it's a 94 degrees Celsius to 96 degrees Celsius is the most common temperature when the DNA get denatured. Okay, so at high temperature, because of this high temperature, that separation of two strands at each strand of target DNA then act as a template for synthesis of DNA. That means of here you can see this is the double stranded DNA. When you transfer this DNA segment into the PCR and raise the temperature up to 96 degrees Celsius or 95 degrees Celsius, what will happen? This double stranded gets separated and it form a two single stranded. Can you see here, one strand over here, one strand over here. So how many total? Two strand get separated as a single stranded. Is this clear? Okay. Now after separating, what is the next? The next is annealing. Annealing means to join. Now how you will join this? For joining you need a primer. So here you can see annealing is the next step after denatured. The next step is annealing. So annealing, at what temperature you perform the annealing? You have to keep your temperature down. What should be the temperature? It should be near about 55 degrees Celsius. From 95 degrees Celsius, then it becomes double single stranded. Then you keep the temperature down, which is near about 55 degrees Celsius. So after keeping the temperature 55 degrees Celsius, what happened? The two oligonucleotide primers annealing to each other, the single stranded template DNA, since the sequence of primers is complementary to the 3' end of the template DNA, and this step is carried out at the lower temperature. And what should be the temperature? As I said, 55 degrees Celsius, but the most common is 40 degrees Celsius to 60 degrees Celsius is the most common. This is the minimum and this is the maximum range. So you can just remember. Okay. So it depends on the length of even this also depends on the length and sequence of primers, that what you are using and what is the length of DNA, that depends. That's why I wrote the 40 degrees Celsius to 60 degrees Celsius is the annealing of primers required this much temperature. So you can see this is the primer. The green one is a primer. So you anneal the primers, means you join the primers over here. So for joining the primers, you need a lower temperature, right? And after annealing the primers to the DNA, the next step is called extension. Now, what is the extension? So the extension means that is polymerizations. So this is the final step, which is called extension. Now here, tag DNA polymerase synthesis the DNA region between the primers, and that can be used DNTP and magnesium. So here use as a D and let me write here, not like that. So let me write it is D and TP. DNTPs plus magnesium. Okay, so it uses DNTPs and magnesium. It means the primers are extended towards each other so that the DNA segment lying between the two primers is copied, and the optimum temperature for this polymerization is near about 72 degrees Celsius. That means of again you have to raise the temperature up to 72 degrees Celsius. So this is a very simple experiment, and through that you can make a multiple copy of your desired DNA. Okay, that's why it is a polymerase chain reaction, PCR. Okay, so here tag polymerase, DNTPs and magnesium is used for extension, and here you can see the new DNA being made. You can see this is the new DNA, this is your primer, Here is your primers. Now here you can see this is your new DNA, this is your new DNA, right? Formation of new DNA. So in this way you can make a multiple copy from this PCR. Now being the second cycle, this is the first, this is just a one cycle, means to complete the one cycle 3 steps are required, and all these 3 steps, like one first step is called what? Tell me the name of first steps.
Denaturation.
What?
First step, sir.
Yes. All good.
Sir, that is the first step, sir. Denaturation.
Yes. Denaturation is the first step. Second step?
Annealing.
Yes, annealing. Then second. A third?
Extension.
Extension. What is the temperature of all three?
Sir, for denaturation it's a high temperature, and for annealing we have to again
High temperature means what is the temperature range? Tell me the range and the exact temperature.
like 94 to 96 is the range,
Yes.
and for annealing 40 to 60 is the range, but we generally take 55.
Yes.
For extension it is 72 degrees Celsius.
72 degrees Celsius. Clear? So this is a very simple procedure through that we can multiply our desired DNA. Now let's understand the application of this PCR. So you can see PCR is used in various their application is very broad spectrum. The most common, anyone can tell me the most common and recent use of PCR? We recently used that PCR for what purpose? And everyone performed this. DNA fingerprinting is not very common. The DNA fingerprinting is generally performed in the case of, yes, see, Sarjana said COVID test. Yes, everyone passed with this PCR techniques, right? So this one is the most common one, the PCR to detect the pathogens. Pathogens, that means of whether you are suffering from COVID-19, yes or no. To detect that, we need to perform a PCR. So it detects the pathogen, very first application. Second, diagnosis of specific mutations. Like suppose any pathogens who become a mutant, so that can be easily diagnosed with this PCR. DNA fingerprinting, no doubt. DNA fingerprinting is also another case where we can use for different type of diseases, like we can say for detecting the crime, generally in a crime scene, forensic laboratory, this DNA fingerprint by using the PCR. Then detection of a specific microorganisms, like any bacteria which is not known yet. So we can, through this PCR we can detect them, the specific pathogens. And even in the parental diagnosis, means we have to find out the genetic disorders in any child. So we need to perform the PCR, so we can able to detect any genetic disorder from the parent. So it's a parental diagnosis can be easily detected by this PCR. So these are the other applications related to except an RDNA technology. Now, so the first we isolated the DNA. Second, we performed the cutting of DNA at a specific location. Third, we performed the amplification of gene of interest using PCR. Now the next step is after PCR, after making a multiple copy of that interest of gene, that we need to preparation and insertion of recombinant DNA into the host cell or we can say the organisms. Like here you can see this is a multiple copy of your gene of interest that you need to cut that by particular segment, and then you have to incorporate with the vector. So restriction enzymes cut out the DNA of interest and open the vectors, and once it open, now you can insert your desired DNA by using DNA ligase, join the two DNA sequence together, and this become a recombinant DNA, or we can say the plasmid DNA, which is DNA of your interest. Now this plasmid you have to incorporate. This is the techniques. DNA ligase is basically helping to form a recombinant DNA plasmids. Now you have to incorporate that into the bacterial cell. Suppose this is the bacterial cell. So in that bacterial cell, you have to again reinsert this into the bacterial cell. And this bacterial cell, now you can grow in a large container, or we can say any media that is suitable for that bacteria. So it will multiply in number. Yes, I will give you break because it's a 2-hour session. So just this is the last one to obtain the gene product, and then I will give you break for 5 minutes, and then we will start our session. Okay. So obtaining the gene product, that means of what? When the recombinant DNA is transferred to the bacterial cell, plant cell or animal cell, the foreign DNA is multiplied. And most of the recombinant technologies aim to produce a desirable proteins. So there is a need for expression of recombinant DNA, right? So for that purpose, we need to be cultured, so that cultured either it can be in container or in any laboratory specific media, so that we can able to extract the desired protein of interest from that bacteria, right? So we can see in the next after the break that how to obtain the foreign gene product. Gene product means how to obtain the insulin, proteins, and or monoclonal antibodies after the RDNA technology. So that we will see after the break. Now, so here 8:33, you all can take a break for 5 minutes, and we will start our session by 8:38. Okay, so you all can take a break for 5 minutes. Hello. Hello everyone. Yes, break is over now. Yes, on your front camera. All of you, please, on your front camera. Okay. So we talk about the obtaining the gene of interest. We completed this part, right? Now come to the, so here you can see there are different how to culture that RDNA, or we can say our desired bacteria to obtain the product. So we require a bioreactor. You can see this is the bigger one. This container is called bioreactor. So in this bioreactor, you can culture your bacteria. Okay. So you can see here bioreactor, or it is also called the fermenters. Bioreactor or fermenter. This is the industrial labor fermenter. This is all the industrial fermenter, like in pharmaceutical or any dairy product. These all are the bioreactor or fermenter. Now this is what is this bioreactor? It is nothing but it is a large vessels in which raw materials are biologically converted into product by the microbes. Means you have to just put all the raw material inside these vessels, and then you put your desired bacteria into it, bacteria or any fungi or any virus into it. Now this bacteria are going to digest that raw material, and they will produce your desired product. So that product by the either can be microbes, either can be plant or animal cell or enzymes. That is called the bioreactor. Clear? Now this bioreactor, you can see this bioreactor, large scale productions that is carried out in the bioreactor. Means if you want to produce large amount for industrial purpose, for any pharmaceutical purpose, then only you can use a bioreactor. If you want to perform at the laboratory level, then a petri plate is enough to culture and perform the experiment, right? So the bioreactor provide a optimal condition for obtaining the desired product by providing optimum growth conditions like temperature, pH, substrate. So temperature, then pH, then substrate, everything is maintained in that bioreactor, like along with the substrate, vitamins, then there is oxygen, because all the organisms require oxygen, even the salt concentrations, everything is maintained in this bioreactor, so that your microbes is going to produce your desired product, that what you are expecting from that microbes, right? So it is going to produce inside this. Now bioreactor can be aerated. Why is it aerated? Because it need a proper aeration, because the microbes is growing inside, right? So if it is growing inside, and if your bioreactor is completely closed and no oxygen present inside, then your microbes may get killed, and that's why it required proper aerated. Stirred for mixing the media. That means of inside there just stir it so that it can be rotate. So for rotation, for shakers, it need a proper mixing, so that media will, or whatever the media present, that is going to be digested on periodically. So they can be made by contamination free, and replacement of used media take place. That can be possible in this bioreactor. Now usually the most commonly used bioreactors is the string types, but bioreactor is of two types. Okay, most common is a string type, but it is of two types. One is called the simple stirred tank bioreactor. Okay, simple stirred tank bioreactor. So here it is the simple types and consist of stirrer which help in the mixing and even distribution of oxygen throughout the reactor. So this is what the stirrer. Okay, so this stirrer is going to sterilize, or we can say it is going to mix all the nutrient equally so that no microbes can grow a huge amount at this left side and less amount at right side. Why? Because there is a stirrer, so it is going to mix properly. Along with it can provide the oxygen. This is called simple stirred tank bioreactor. Another type is called the sparged stirred tank bioreactor. Now, what is the difference in that? The second type, in it, air is supplied in the form of air bubbles. What? Air is supplied in the form of air bubble. That is the only difference. The surface area for oxygen transfer is increased. You can see this is the surface area, so oxygen transfer is maximum on this because it is in the form of air. So that is sparged stirred tank bioreactors. Now you can see this is fermentation of the raw materials is carried out in the bioreactors. So two types of fermentation can take place, namely batch fermentations and the continuous fermentations. Now here this is quite interesting. Fermentation take place by two methods. One is called the batch fermentations and the another is called continuous fermentation. Now what is mean of batch fermentation and continuous fermentations? So you can see here in this, when we say batch fermentations, okay, so fermentation nutrient raw materials are put in the closed chamber and are not changed once the fermentation start. Like here, before start the fermenters, you need to put everything at the same time, like the nutrient, or if you want to put any kind of vitamins, raw material, everything you have to keep ready. It's similar to like when we make dal in cooker. You know dal? Hope everyone remember the cereal. Yes. So for making dal, what we need to do, everything like grains, water, then turmeric, salt, and all the spices which we need in our dal, then everything we have to put inside, and then we close and put on the gas, right? So this is what the batch fermentations. So that means of nutrient and inoculums are added into the cultured vessels before start the bioreactors. And once you start, you cannot open it, and it is going under the process, so nothing is added and removed during this process. You cannot do anything add or anything remove from this bioreactor. And after the end, you will get directly the end product, and that product you can take for the downstream processing purpose. Clear? This is called the batch fermentation. Another one is called the continuous fermentations. What is continuous fermentation? The nutrient and raw materials are added continuously and the product are removed as they are made. Like here you can see, so sterile media reservoir over here, valve is controlling valve. Okay, so you have to add the nutrient from here, nutrient, or we can say vitamins, anything if required you can add, and it is continuously going to produce your desired products. So here one thing is common, that in the continuous process you have to supply nutrient or any required media continuously, and you can withdraw the product continuously. But in case of batch fermentation, you cannot put anything continuously, you cannot withdraw the product continuously from. That is the only difference in the batch and continuous fermentation. Is this clear or quite difficult to understand? Anyone? Okay. After collection of your product, after the fermentation, what is the next? The product need to purify, which because this product is in the form of impurities, lots of unwanted product is also unwanted, what we can say, the material is also present in that product, right? So we need to separate them, because we are interested in insulin, but apart from the insulin some other chemicals may also produced by the bacteria that we don't know, right? So for that we need to do a downstream process. And what is this downstream process? Separation, purification and formulation is a downstream process. Which, and formulation is what? It is the last step of your product. And that product you can launch in the market. So after the product has been made in the bioreactor, it need to be processed before it is marketed. So you cannot directly market that product which is removed from, which is obtained from the bioreactor, like here. Okay, this cannot be marketed because this is untested product. It is not a purified product. It is having lot of impurities, and if you sell in the market, patient will complain and your company license will get cancelled, right? So that's why you need a downstream process, that is separation, purification and formulation. So the process includes separation, purifications of the product before it marketed, and it is together known as a downstreaming. Okay, now separation, purification, formulation take lot of times. I mean to say, it is not included in your syllabus of NEET, but still you should remember what is mean of downstream process, separation, purification and formulation. That is important for you. Now after the downstream processing, many clinical trials, after once you made the product, once your new tablet or new vaccines is ready, you cannot launch in the market. You need to do a clinical trial. You know how much time is required for clinical trial? Near about 7 years. 7 years. Initial 3 years, then 3 years, and then 2 years. And then your product will get approval from what? From where? Tell me. Indian Council of Medical Research. It is a government organization body who give a permission that your product is certified. So it is ICMR. How many of you know the name ICMR? What is ICMR? It's a Indian Council of Medical Research. Yes or no? Tell me fast. How many of you know about this ICMR? No one? So this ICMR is going to give a final approval of your product, and that product you can sell in the market. Otherwise, you cannot sell it in the market. Yes. Sasidharan.
Yes, sir. It is
Yes.
Indian
Ask.
Council for Medical Research.
Yes.
Indian Council for Medical Research.
Yes. It's the Indian Council of Medical Research. So it generally grant your product to be sell in the market. right? Especially for clinical, for medical purpose, for medical field. Like you might have seen that Baba Ram. How many of you have heard about the Baba Ramdev? Ramdev. Anyone know about him?
So Patanjali,
Yes.
what?
Patanjali claimed that they invented a kind of syrup which is called coronil, and from that coronil, corona will get corona infected patient will get cured from that coronil. Yes or no? This kind of something news in past, I think so in 2021 during the corona influence, right? How many of you have heard this news? At least no one. Come on, tell me. Why all the sitting silent? No one is talking. Is something new which I said? No, no, right? Okay. Well, if you're not interested to discuss on this topic, no problem. But you should remember ICMR is Indian Council of Medical Research who certified the product which is good for human beings, and they give a license to sell in the market. Okay, so it is ICMR. So yes, so after the downstream process, it requires many clinical trial. Many means what? It requires, see, corona vaccine developed within a 3 years, or we can say within a 2 years, because corona vaccine lot of AstraZeneca and all get approval in a emergency cases. But for any product, if you want to launch in the market, you need up to 7 years of clinical trial is required. Without any clinical trial, you cannot launch your product in the market. Clear? Because you need to take approval from the ICMR. So this is what, how any medicine, any product, any vaccine, any antibiotics, or we can say insulin is produced. It is produced through this complete process which we studied today. Yes? So this is all about the today's topic. Now anyone have a doubt? Tell me, anyone have a doubt? Okay. No doubt? All clear? Anthony, Prasanthi, Sasidharan, and Sarjana, Gopinath, Ragul. Okay. So let's try this question. PCR and restrictions fragment length polymorphism are the method for study of enzymes, genetic transformation, DNA sequencing, genetic fingerprinting. Okay, so Sarjana responded genetic engineer fingerprintings. Okay, I want response from all of you. Okay, so Anthony said D, Sasidharan D, not sure sir. Okay, why not sure? See, I know RFLP is new for you. RFLP, which is called restriction fragment length polymorphism, and PCR is we just studied. So PCR is polymerized chain reactions, which amplify, which basically used to multiply the desired DNA, right? Segment. Yes. So, and restriction fragment length polymorphism. See, the name itself, length polymorphisms, means it is going to separate the DNA according to their length. So what actually we are doing here? We are using, we are multiply the DNA by using PCR, and we are going to separate according to their size. So if the DNA is going to separate according to their size, it is come under the sequencing, because you are sequencing the DNA, right? So if you are sequencing, then it is a part of DNA sequencing. It is not a genetic fingerprinting. This is talking about the method. It is not talking about the use of PCR, and that's why this is the wrong statement. Study of enzyme cannot be possible for this case. And genetic transformation means what? Are we going to transfer the genetic information from one segment to another? No, we are just multiplying the number by using PCR. So this is also not the right way. So the DNA sequencing is most common method. So C is the right answer. Those who selected D, that is wrong. C is the right one. And make sure next and also. Okay. And the right answer is DNA sequencing. Is this clear to all of you? Okay. During PCR techniques, the pairing of primers to single stranded DNA segment is called. I think so. This is very simple. Single stranded. SS means, here SS written means single stranded. Okay, keep in mind, SS DNA means single stranded DNA. So when you add this single stranded DNA along with the primers, and when the primer is pairing with the single stranded DNA, this process is called annealing. Now this is sure. Yes. So it's an annealing process. Sajana, why you not responded? Sajana, any doubt? Then why you not responded?
Sir, actually I was about to respond, sir.
What?
I was about to respond, sir.
Okay. But forgot to respond. Right? Okay, alright, no problem. Let's move ahead and see the next question. Okay, another question, and I want this answer from Prasanthi. Let me recall the name, and I want a response in the chat box. Sasidharan, Gopinath, Prasanthi, and Ragul. Please respond this question's answer in the chat box. Prasanthi, hope I am audible to you. Yes, Prasanthi? Am I audible to you? Okay. See, Gopinath very nicely responded. Gopinath, Prasant. Okay, Prasant responded first. Okay, Sasidan responded D. Okay. Question is, an analysis of chromosomal DNA using the southern hybridization technique does not use blotting autoradiography, PCR, electrophoresis. Oh my god, this is also something new for you. Southern hybridization techniques. But I think so. This is also simple. And said it was in the DNA fingerprinting of fifth chapter, sir, molecular basis of inheritance. Okay, okay. See, there are here it is mentioned as southern hybridization, southern, northern, and western. Please remember southern, northern, and western hybridization. Hybridization. Southern is for DNA, DNA. Northern is RNA, and western for protein. Okay. Now, when the topic is an analysis of chromosomal DNA using the southern hybridization technique does not use blotting, autoradiography, PCR, electrophoresis. The most right answer for this is PCR will go, because remaining all the parts of hybridizations, blotting hybridizations, autoradiography, right? But PCR is not the part of this. So yes, definitely C is the right answer, and this is the wrong one. Because southern blotting is a laboratory method which basically detect the specific DNA molecule among the mixtures of other DNA molecules by using technique like electrophoresis, autoradiography. And this method reveals the information of DNA identity, size, and abundance. But it does not use the PCR or PCR method to use the amplification of DNA sample. And that's why PCR is the right answer for this question. Another one, okay, so this is the summary what we studied today. We studied about the polymerase chain reactions, the most specific instrument which is discovered by the Carey Mullis, name of scientist Carey Mullis, who invented these techniques. And this polymerase chain reaction is in one complete cycle, one single cycle required denatured, annealing, extensions, and amplification.
So,
Yes.
sir, it is tagged DNA, bro, right, sir?
Yes, it's a tag, not tag, it's a tag, TAQ.
They have mentioned it as tags.
Here, right now?
Yes, sir.
Okay, just make a corrections. This is a tag.
Okay, sir. And what is DNTP, yes, sir?
See, DNTP, using DNTP, that is dinitro triphosphate, is a kind of, we can say, the enzymes that is going to use, at the phosphate group, because to break the phosphate group, it required energy, right? For that purpose, there is a using DNTPs, it is going to break and it release the energy so that it can work. So basically, this tag polymerase help in the synthesis of new fragment. How it is going to synthesize? By using the TDNTP.
Okay, sir. So,
Clear? Yes.
Sir, can you give a gist about amplification, sir? Like that time I lost my connection, sir.
What you want? Amplification?
Can you give a gist about it, sir? Like, can you?
What is amplification?
Yes, sir.
Okay. See, simply, if you're asking me about the amplification, it is what? This is the part of PCR. Okay. When we say amplification, means you want to multiply your gene of interest using PCR is called amplification, right? Like suppose you isolated DNA and your gene of interest is just a single segment, like you have only this much segment is your gene of interest. Okay, this is just a single one. Now you want to multiply this single gene of interest in n number. So for that purpose you can do a amplification. So the desired DNA fragment is amplified around billion times, means you can multiply your interest, your desired gene billion times or n times. We can say like suppose you want to multiply 200 times. So you can, from the single copy, you can create a multiple copy with the process that is called amplification by using PCR technique. And PCR is always follow a 3-step cycle that is called denatured, annealing and extinction. Clear now?
Yes, sir. Clear, sir. Thank you,
Got
sir.
doubt?
No, sir.
Yes?
Clear, sir. Clear, sir. Thank you, sir.
Good, good. So this is about the PCR. Now here you can see the steps which we studied, that how to isolate, or we can say how to multiply the desired vectors into the bacterial colony. So the first step is you have to isolate your desired gene, that gene of your interest, like you want to isolate insulin or you want to isolate some vaccines. So you have to first select the cell, and from that cell you have to extract the DNA, and you know the steps, the process of how to extract the DNA, as I told you, for extracting the DNA, because when you extract the DNA, it is not coming just a DNA, but apart from that, some other macromolecules will also come together with the DNA. So we need to separate them. So for that purpose, we need to use a different enzymes to separate. Like suppose if DNA along with the RNA is there, to remove the RNA, we need to use RNase. To remove the protein, we use protease enzymes, right? So that we have to use to separate all the macromolecules. Your desired product is only DNA, so you can just select that DNA. You can make a cut by using a restriction nucleus enzymes, and that cut you can insert into the bacterial plasmids. It become our recombinant plasmids, and these plasmids you insert into the bacteria, and when this bacteria multiply, you will get n number of resistant bacteria which is capable of producing your desired gene. Here, so this is how the complete process of RDNA technology that we have studied. We also studied out the downstream process and bioreactors and all, so that is very basic, so you know very well.